what we investigated is the cause of death of the children of Australian Kathleen Folbig, who was in prison for 20 years, accused of having killed her 4 children. We discovered a novel genetic variant in the gene CALM2 that encodes for a protein called calmodulin that regulates the heartbeat. And we were able to show that this particular mutation caused arrhythmia, irregular heartbeat, that was likely the cause of the death of the daughters. And eventually this led to her exoneration in 2023. We had been establishing a pipeline to identify rare genetic variants that could explain autoimmune diseases in children so that we could understand mechanisms. And with that, we had the tools to discover potential variants that, could be important. And I received a phone call from a lawyer that had done an honors degree in my department. And, he said that, there might be a genetic cause in this family. He had struggled to find anyone interested to help. From a research perspective, I thought I could help. So I ended up, doing the sequencing in the mother, who eventually had the variant that eventually we found in the children. When I started working in this case, I never expected it was going to turn into a 5-year long journey, 2 legal inquiries, hearings, many reports. Yes, it was a hard time. It was challenging. We met quite a lot of problems and obstacles, but it's also been enormously rewarding. I think as a scientist, to be able to do something that changes, even if it's one person's life, it's very special. So I don't regret having done this work. I'm happy I got involved, but it did take a lot of nights sleeps and it was quite difficult at times. Invitation originally was to participate in this students training course, the beginning of a PhD. I think it's a very important part in the career of a student. I like doing those things. I liked chatting with students. It was a wonderful talk. I think you've got actually a very, very talented cohort of students and quite mature as well.